Hi, I'm Jim Walgenbach, Extension Entomologist with North Carolina State University. I work in the area of tree fruits and vegetable crops. And this year we've had a little bit of a challenge with the pandemic, and I was concerned when we first started the year that we might not have much of a research program this year. But we've been fortunate, and we've been able to carry on a pretty good field research program this year, particularly as it relates to two of our key insect pests on tomatoes, that being the two-spotted spider mite and the western flower thrips. What we'd like to do today is give you a brief overview of some of the work we're doing with these two pests, and in the process, hopefully we can introduce you to some of the key people that are doing the work outside of myself. Thank you.
Hi, I'm Tom Bilbo. I'm a postdoc researcher in the Walgenbach lab. I manage a number of research projects improving the management of spider mites and thrips in tomatoes and other vegetables, specifically enhancing the biological control of these pests. We have a number of research projects on NC State research stations in the mountains and in the Piedmont, as well as on a number of commercial farms, thanks to a number of cooperating growers.
The two-spotted spider mite is a key pest of tomatoes in North Carolina. Their feeding on tomato foliage reduces overall yields, and at high densities, they can feed directly on fruit, resulting in a cosmetic blemish known as gold fleck. The traditional approach to managing spider mites in commercial tomatoes is through multiple applications of acaricides, but this reliance on chemical control has a number of limitations and drawbacks, including a period of pest suppression of only a couple weeks, high costs of acaricides, resistance development, and harvesting limitations such as post-harvest intervals of up to 7 days. To improve pest management in tomatoes, the Walgenbach lab has been developing a biological control system that releases predatory mites into tomato fields for long-lasting spider mite suppression. Phytocoelis persimilis is a highly effective predatory mite that specializes on spider mites. Over the last several years, we have been conducting experiments in research trials and on commercial farms to determine the best ways to release this predator. This predatory mite is most effective when released at a rate of 20,000 per acre, and when spider mite populations are first establishing in a field, approximately one mite every other leaflet. One example from a commercial tomato field demonstrated that persimilis released into a 0.125 acre section of a field kept spider mite populations low in that release section, but also that persimilis quickly increased and dispersed and virtually eliminated spider mites from the entire 2.5 acre field by 5 weeks after predators were released. Ongoing research is being conducted to determine how widely persimilis disperse and manage spider mites after being released into fields, especially in larger fields. The Walgenbach lab is also researching another method of utilizing persimilis for spider mite management in tomatoes. These predators are released into adjacent or intercropped beans that are infested with spider mites. The beans serve as a production facility for persimilis, where they increase and then disperse into the nearby tomatoes. In addition to releasing persimilis into fields, natural populations occur in some parts of North Carolina. These populations can occasionally provide effective spider mite management later in the season. However, only if certain pesticides toxic to this predator are avoided. We have ongoing research to enhance the presence and benefit of these natural populations. Please visit the Walgenbach Lab website for more information on using persimilis and for a list of compatible pesticides. Another key insect pest of tomatoes and other vegetables is the western flower thrips. This insect can feed on foliage, reducing yields as well as causing injury to fruit such as gold fleck and dimpling. Western flower thrips can also transmit a number of pathogens, such as tomato spotted wilt virus. To better understand and manage this pest, we have been monitoring thrips populations in the mountains and Piedmont in crops such as wheat, tomatoes, and peppers. These studies allow us to better understand how different thrips species compete within crops and move between crops throughout the growing season. In tomatoes, we monitor for the eastern and western flower thrips by collecting flowers. Identification of thrips species can be difficult and requires a microscope to see key features. Additionally, we collect western flower thrips populations and establish colonies in our laboratory. With these colonies, we are able to conduct insecticide bioassays to monitor for resistance. In recent years, we have detected field resistance of western flower thrips to radiant in multiple locations in Rowan County, and are currently monitoring for changes in susceptibility to beleaf. We are also exploring improving the biological control of the western flower thrips using the predatory mite Amblyceus swirskyii, and research studies are underway.
Well, hopefully you've enjoyed learning a little bit about the work we're doing with spider mites and thrips. And one thing I'd like to point out is our whole goal during this entire process is to make sure we're doing our part as entomologists, to make sure that we're producing fruit, or producing recommendations that allow growers to produce beautiful, blemish-free fruit that can make growers profitable throughout the growing season. And I'd like to thank the growers that have been cooperating with us in much of our field research, as well as the people you've met during this video.